On behalf of Friends of the Lost Coast and the Bureau of Land Management King Range National Conservation Area, welcome to Lost Coast Lectures. My name is Justin Krellin, Executive Director of Friends of the Lost Coast, and tonight we host Sarah Conway, botanist with Redwood National Park, in her talk, Introduction to the Wonderful World of Lichens. Thanks for joining us, Sarah, and please share a little bit about yourself and your journey as a lichen researcher before you get started. Take it away.
Thanks so much, Justin. Hey, everyone. Thank you to the Friends of the Lost Coast for having me. I see some familiar names out there. It's great to see everyone. So, I started my lichen journey in a kind of roundabout way. I grew up in South Carolina and originally went to school for English, and I minored in hospitality just because that was a practical thing to do. And my husband and I ended up moving out to Humboldt about 12 years ago. And I was still working in hotels out here, but I just kept meeting people that had really cool jobs. I met people who were watershed managers and a woman who studied salamanders. And I just remember asking them, like, cool, but what do you do for money? What's your actual job? I didn't understand growing up that one could be a botanist or could be a land manager. So I ended up doing kind of an about face out here, and I went back to school to get another undergrad degree in botany at Cal Poly, back when it was still Humboldt State. And I got a summer job working for Redwood National Park as a botany technician on the vegetation management crew. So, treating invasive plants, monitoring rare species, doing compliance work for the park. And I just completely fell in love with the work. And I ended up going straight into my master's degree at Cal Poly when I finished studying biology. And I did my thesis work on lichens. I did a giant lichen inventory of the Horse Mountain Botanical Area. I ended up collecting almost 900 specimens, and I ID'd all of them to species, and I did DNA sequencing on them, and I was able to describe a new species of lichen, which we'll talk about. So I really just fell in love with it, and I am still working at the park. This is my 8th year up there. I now am the lead botany biotech. And I just incorporate lichen work into our biodiversity monitoring at the park as much as I can. And I also have been able to do lichen contract work for the Forest Service all over California, doing lichen air quality monitoring, which we'll talk a little bit about. So that's who I am. I'm so happy to be here, and I am excited to share lichens with you. I realized today that this is the first talk that I've given that isn't an obligatory talk for my research or a project that I've been working on. So this is my first just kind of fun lichen talk. The wonderful world of lichens. Uh, like I said, I'm excited to be able to just kind of talk about what I want to talk about. So this can be open-ended. If you have questions, feel free to interrupt. Like Justin said, raise your hand or put a question in the chat. And then at the end, we'll have time for a Q&A as well. But for now, what I want to talk about is the green blur. Some of you may be familiar with this concept. In natural resource management, we talk a lot about the green blur. It's basically referring to the perspective held by the vast majority of the population, unfortunately, who tend not to notice the diversity of organisms around them. They may go outside and notice the blue sky and then kind of a blur of green where all the plants are, and then some brown stuff on the ground. And that is about the extent of their outdoor experience. A beautiful tree might stand out, or a brightly colored flower. But in general, if you're looking out into a field of greenery, that's all you're seeing is greenery. Uh, you all have already broken through the green blur a bit, right? You're attending a lichen lecture on a Thursday night on your own time. So you probably know a few species of fungi or birds or plants, if not many species. When you go outside, you recognize a few of those species more and more as you go along. You probably started off by learning a few of the most kind of charismatic species, and then over time, You start to see them everywhere. When you travel, you're noticing familiar species. You start to expect particular species to show up when you're in different habitats. And when you run into species that you recognize, to me, especially when I'm in a foreign place, it feels like I'm kind of running into an old friend. And it just really enriches your life. When I think about it now, it's hard for me to imagine a time when I didn't notice lichens. Uh, but as I'm about to explain, it wasn't entirely my fault that I didn't notice them. So, neuroscience has shown us that our brains are always optimizing for efficiency, right? The brain is going to alter sensory input to make perception easier or faster. So it's creating shortcuts, it's strengthening pathways based on like predictive coding to guess what is next. So it's your brain is filling in gaps to kind of simplify complex information or fit that information into existing understood patterns. For example, look at this little image that I have pulled up. If you're looking at square A and square B, you're likely seeing that square A is much darker, square B is the lighter shade. But what's really happening here is that your brain is using a pattern that it understands about shadows, the shadow cast from the cylinder here, and it's adjusting those colors to make it make sense to your patterns that you recognize. So in reality, square A and square B are the exact same color. Your brain is simplifying the world around you and it thinks it's helping you, but I'm here to tell you that you're actually missing out. So learning about lichens and bryophytes actually changed the way that I experience the world. And that sounds like a big claim, but it's really not. Uh, every time I took a class at Humboldt, I felt like I was adding new lenses to my eyes. Like I could suddenly clearly see an entire host of organisms that I was just glazing over before because my brain didn't recognize them. It's just smoothing them over. I know what tree bark looks like. My brain just kind of smooths the bark over and I don't see the detail. I remember clearly the first time that I went back to South Carolina after taking a bunch of mycology courses out here. And it was truly mind blowing, the amount of biodiversity that I had in my own backyard. And I had considered myself, a fairly observant outdoor kid. So to have just not seen all of the fungi back home because my brain was just glazing over it was really wild to experience. But now I have these new lenses and everything is more interesting. So that is my goal with you all tonight. I hope that we can add a lichen lens to your eyes if you don't already have one. This is just a quick example of blurred diversity. This is my front porch. That is my railing. Not very exciting. You might look at it and see that there's a little weathered look to it. But if you look closely, there are tons of species of lichen covering every square inch of wood. Um, you can see these little structures here. That's a lichen species. These little white structures are also lichen. This yellowing is lichen. In fact, these black lines are actually created by the lichens. They're kind of they're delineating different species. So it happens when two different individuals come up against each other. And it's because lichens produce a whole suite of chemical compounds that are unique to lichens, like thousands of them. And so when these chemicals meet, they kind of form this black, dramatic border, like on a map. Really cool. Another benefit of getting into lichens, you don't have to go far to find them. No backcountry hiking required. Again, this is my back porch. Rocks will look like works of art when you're clued into lichens. There are many, many benefits. Okay, so you are ready to conquer the green blur, but why do we bother with the lichens? There are lots of aspects to the blur that you could be clearing. So why are we bothering studying these tiny little critters? Well, I'm going to first make that case to you by explaining a few of their ecological roles. Then we will get into detail about, what exactly is a lichen. I will go over some lichen vocabulary and morphology. And I will also talk about methods for identifying lichens to species. So I'll introduce you to some species that you can ID on your own here in Humboldt and Mendocino without chemical testing or without microscopes. And then I will also leave you with some resource recommendations. Oh, and this, I'll introduce you to our little local friends here. And then those resource recommendations will come at the end since you'll all be just jonesing to get out there and look at lichens. So for starters, lichens are vital nutrient cyclers. They are able to uptake mineral nutrients directly out of the air or through fog and rain deposition. So many species are able to fix nitrogen. They hold onto these minerals that would normally just be washed away through runoff, and they allow them to be held close to the tree or whatever its substrate is to kind of slowly drip release these minerals into the immediate environment. Lichens are also primary successionists, which means that they are the first organisms to establish themselves on newly exposed land. Think of a newly formed volcanic island. The only way for that landmass to eventually be covered in vascular plants and therefore be hospitable to wildlife is for lichen to colonize the rocks, which would provide some kind of surface area for the buildup of soils and so on. Lichens are also tools by which we can monitor air quality, as many lichen species are very sensitive to certain air pollutants because they're uptaking, like I said, those nutrients right out of the air. In fact, here are two figures, pretty interesting, that show this region in Italy where a study was done on air pollution and its impact on death from lung disease. On the left, we have a map of lichen biodiversity. And on the right, we have a map of mortality rates from lung disease, nearly identical. There is a short Nature article about this if you're interested. It is Sislagi and Nemes from 1997. You can see here in this area, very low species diversity of lichens. The lichens are not very happy down here, and that is the highest area of lung cancer mortality, just as these areas have very high, very happy biodiverse zones of lichen and very little lung cancer. Pretty striking. Lichens are also essential for many of our favorite charismatic species. Elk, caribou, other ungulates use them as winter forage. Birds and tree voles use them in their nests. You can see this hummingbird's nest is just covered in lichens. And they use them in nests because they have excellent antimicrobial properties, so they help keep the nest clean. Flying squirrels, like our Humboldt flying squirrel, relies on this lichen here. It's called Briaria for up to 90% of their winter diet. So, who depends on there being plenty of flying squirrels to eat and plenty of tree voles? People like our beloved and endangered spotted owl. So, management practices that affect lichen populations will secondarily affect these organisms. Something to keep in mind. Lichens also provide habitat and protection for countless invertebrates who feed on their little protein-packed propagules and use them for camouflage, even live inside of them, as is the case for these little mites and springtails. I can't tell you the number of times I have been poking around at a lichen under a dissecting scope, and I've just been met with a set of eyes just like this. There are so many adorable jumping spiders living in lichens. I've also seen countless turrets from our native California turret spiders that have been built with lichens. Do you know about turret spiders? I'm going to put the genus into the chat. It's called Adapoides reversey. Such a cool spider. They are our only trapdoor spider in Northern California. And they build these really cool little towers out of whatever natural material is around. And they're just so interesting because they always look different. They're never built the same way. They always use different materials. And they build these little towers with a web inside, and they wait near the top for an unsuspecting insect to crawl over the natural material, shaking the nest, and it pops out to get its prey. One of the coolest things about these spiders is that they can live for 16 years, which for a spider, it was mind blowing to me. They're really small too, tiny little brown spider, 16 years at least. And actually, once you start finding these turrets, you can look around them, and often there will be even smaller turrets nearby that are the family members of the other spider with the larger turret. They make little neighborhoods. It's adorable. Here's another turret that was built with lichens. You can see I found this up in the park. And then on the left, we have a couple of examples of lichen camouflage. This is a moth up top here. And then this is a spider. You can make out the body and the legs here. There are so many examples of really cool lichen camouflage. I highly recommend looking it up on your own time. It was hard for me to narrow down. I mean, I didn't narrow it down too much. There are several more examples. This is a katydid that looks just like this Usnea species, wild. This, some kind of little mantis thing, I think. It looks just like a Parmotrema species. And then I actually took this picture near Cal Poly of this gorgeous lichen orb weaver. And you can see that it's trying to mimic this Parmelia species right here. And it even has not only the color exactly, but just the shape of the lobes. Really, really cool. I never would have seen this beautiful spider if I hadn't already been focused on the lichens. I got to go to Tanzania last month on a mycological expedition. Don't have time to get into that now. I'll be giving a talk in September for the Mycological Society of Humboldt. Keep an eye out for that. But when I was there, I was obviously checking out the lichens as well. And I saw this little weirdo crawling around on a rock. And it's a little jumpy, but it's basically an insect larvae. It reminds me of a caddisfly. It's made a little house out of lichens, either out of lichens directly, or it's just able to really mimic them incredibly. It reminds me of the little caddisfly larva and the houses they build out of the river pebbles. And then I saw this walking stick when I was on a different mycological expedition down in Guyana in South America. And at first, I thought that it had just kind of placed lichens on its body to help camouflage itself. But then, upon further investigation, I realized that it was just part of the walking stick. And as someone who already was really familiar with lichens and looking at it with my hand lens, it was incredible just the accuracy of this camouflage here. You can see it walking around, all those little green splotches on its joints, like I said, just are identical to lichens. And it's just a lot of time evolving. Really, really cool. Okay, so we can all agree now that lichens are cool and important, right? But what exactly is a lichen? I'm going to be introducing a lot of lichen vocabulary here. There's no real way around it to talk about this stuff. My recommendation to you is not to take screenshots or photos. I mean, you're welcome to if you want to, but in order to learn this stuff, it's really helpful to write it down. Grab a sheet of paper, grab a notebook and a pen, and write down some of this vocab. And then, like Justin said, this will be recorded and posted on Friends of the Lost Coast YouTube channel. So don't worry. If you missed anything, you can go back and revisit it if you want to. Okay, so lichen is short for lichenized fungus. You may already know that fungi are heterotrophic, just like us. Unlike plants, which are autotrophic and produce their own carbon through photosynthesis, fungi cannot create their own food. They have to seek it elsewhere. Some fungi have evolved to, break down dead, decaying matter. Some have evolved to form mycorrhizal relationships with trees, for example, where the tree is providing the carbon. Um, Lichens are very similar to mycorrhizal association. They enter into symbiosis with another organism in order to obtain that carbon. So the lichen symbiosis is made up of two or more partners, or symbionts, we call them. The mycobiont, which is the fungal partner, and the photosynthetic partner, which we call the photobiont. So the mycobiont is the fungal partner. The photobiont is whatever that fungus is using to photosynthesize and get carbon. Now, the fungus is obligated to the lichen symbiosis. It cannot live without its little photobiont partner. The photobiont, on the other hand, is a species of free living green alga or cyanobacterium, sometimes both. Oh, yeah, so obligate fungus for the mycobiont. The photobiont, like I said, free living green alga or cyanobacterium, sometimes both. So the photobionts have lives entirely outside of the lichen symbiosis. Here is a photo of one of these photobionts, Trentipolia. And so these little cells are just floating and they're in the air. They'll be on signposts, fence posts, tree bark, anywhere the environmental conditions are right for them. It's only when they are captured by the mycobiont, by the fungus, that they enter into this lichen symbiosis and thereby alter the body plan of the fungus into the lichen thallus or body. That is the word for body, the lichen thallus. So there are not even 200 described species of the photobiont, of the algal partner, but there are roughly 20,000 described species of mycobiont, the fungal partner. So there's a lot of overlap by the photobionts where they can enter into successful symbiosis with hundreds of different mycobiont species. So it would be really taxonomically, naming the species, it would be really confusing to try to name them based on the photobiont because the photobiont mixes with a lot of different mycobionts. So when we talk about a species of lichen, we're actually referring to the species of mycobiont, the species of fungus. Up in the top corner here is a nice cross section of a lichen thallus, aka body. And you can see this white spongy layer of hyphae. And then the fungal hyphae are kind of like the strands of fungal cells. They're very stringy. And you can see this bright green layer here is the algal layer, the photobiont that the fungus is containing within its body. So the fungus ends up controlling the photobiont's reproduction. And once the photobiont is captured, it's pretty much a permanent part of the symbiosis unless it's stolen by some other lichen species. These are really incredible SEM scanning electron microscope photos of a common lichen. I did not take these. These are done by the Eye of Science. Um, this common lichen is Parmelia sulcata, and they have colorized this so that you can see the green photobiont cells. What they're doing here is forming a network of nutrient exchange. So you can see that these fungal hyphae, all these little strings, part of the fungus, have just wrapped around these algal cells, forming these sheaths. And those sheaths are these networks of nutrient exchange. So they can uptake carbon from the photosynthesizing cells, and then in exchange, the fungus is providing the photobiont with protection from the outside elements, water and mineral nutrients that the fungus can absorb directly from its surroundings. Some people get philosophical here and argue that the lichen is, controlling the alga, and so it's not an equal symbiosis. But I like to think that all of the partners are happy. Here at the top, you can see the skin of the lichen. More vocabulary incoming. This is called the cortex. Then we have that photobiont layer. In this case, it's an algal layer, green algae. And then in the center, we have this medulla, which almost looks fluffy because it's just made up of the loosely woven hyphae of the thallus. If you go outside tomorrow and you rip open a lichen lobe, you'll likely be able to see this white inner medulla. Many lichen species also have a lower cortex. All of these morphologies are necessary for identifying lichens or getting anywhere in a lichen key, really. I should also mention that this is really the simplest definition of a lichen. We are learning more about the symbiosis all the time, but we have still yet to successfully grow a lichen in vitro in the lab. We don't fully understand all the components of these composite organisms. We now know, thanks to molecular work, that there are often secondary fungi present in the lichen thallus. And we believe that the microbiome is playing a significant role in thallus assemblage. So there are still a lot of unanswered questions for any budding lichenologist out there. So in the top right-hand corner is that lichen I was talking about, Parmelia. Actually, let's just go back. Take a look at this. Picture one of these lobes being sliced open, a little cross section. And that is what we were seeing in these scanning electron microscopy photos. This is that same species of lichen, Parmelia. So that is what's going on in the interior of those lobes. So there are a few main body plans that you need to know to begin identifying lichens. More vocabulary. Here we have a fruticose lichen on the left, which instead of being flattened like a leaf is kind of shrubby and branched. Some of them are pendant, like this Ramalina here, but others are more erect and kind of bushy. This leaf-like body plan here of the Parmelia is known as foliose. I like to think of foliage. It has a distinct upper and lower surface. And then we have squamulose, one of my favorite words. It is referring to these tiny overlapping lobes. So it's kind of a disjointed thallus or body, not one big cohesive piece, but lots of little overlapping lobes. And you can see them at the base of these cladonia cups here. And then finally, we have crustose lichens who are oppressed or like flattened against their substrata. Many of these crustose lichens are actually lacking that lower cortex that I talked about, that lower skin, and they're instead, the hyphae are directly embedded in the substrata. When this is the case, when they are embedded, that is when they are referred to as microlichens. You'll hear, and if you're buying literature, there'll be books that are like the macrolichens of the Pacific Northwest or the microlichens. So here's that distinction. And it's not as simple as small lichens and big lichens. This is an example of one of those microlichens that would be embedded in the surface. And then here is an example of one of my little specimens. And plant people in here may recognize this as the tip of Calicetus decurrens branch, California incense cedar. The entire thallus is only a couple millimeters wide. It's super, super tiny. But it's not embedded in the scales of the incense cedar. I can just peel it right off. So it's not a microlichen. Even though it's super, super tiny, still technically a macrolichen. Most microlichens require compound microscopy for identification. So you'd have to have a compound microscope, and many of the keys, the dichotomous keys that you would use to get to species, start like the first key step is spore characteristics. So you have to isolate spores from these little critters before you even begin to identify them. So much more complicated than macrolichen ID in general. So you might be wondering how professional lichenologists go about surveying for lichens when it often looks like this picture out there. This is a picture I took on Horse Mountain. And now that we know also how tiny they can be. But luckily, the US Forest Service already has a protocol in place in which you can just select 1 acre circular plots. I'm mentioning this mostly because if the Friends of the Lost Coast ever decided that they wanted to do a little bit of monitoring in the area, they could set up an informal plot system like this to do some lichen monitoring. Just a thought. So this FIA protocol from the US Forest Service stands for Forest Inventory and Analysis, and it's used to monitor vital signs for forest all over the US. They have tons of plots in North America, and each plot is a circular acre, and they measure and collect data from a bunch of different organisms. I, of course, was only focusing on the lichens, and so this is how I would approach surveying an entire acre for lichens according to this protocol. And of course, there are a bunch of facets of the protocol that I don't want to get into tonight or have time to get into, but basically it dictates that I have a time limit of 2 hours. So I'm sinuating through the plot over the course of 2 hours, and I'm looking at only epiphytic macrolichens. So macrolichens growing on other plants. And I will look from a certain height above the ground to as far as I can reach. So there is a nice little kind of parameter of focus. I will be collecting lichens in these little paper packets, and I've got my hand lens out, and I'm closely inspecting as I go around for variations in color, morphology, substrate. You may still be thinking that this is like an impossible task for an entire acre to be inventoried in 2 hours, but you would be surprised how quickly your brain starts kind of picking up on these patterns of those body plans that we talked about and the subtle nuances in color and texture. Those of you who have done any botanical surveys know how quickly you can start to kind of pick apart vegetation as you cruise through a plot. And this is pretty much exactly the same, just on a slightly smaller scale. Uh, I would collect every species or morphospecies, species that kind of looked alike that I couldn't tell apart, that I would notice during that time, during my 2 hour time limit. I'd put each one in a paper packet and write down, details about it, the substrate that I found it on. And the substrate is important because certain lichen species can only be found on certain substrates. So some are only found on rocks or particular types of tree species. It's mostly all related to the pH of the substrate and the ecological preferences of that lichen. So, for example, redwood trees tend to only have a few macrolichen species, and that's because the bark is highly acidic from all of the tannins. You compare that to, say, a Doug fir immediately next to a redwood, that Doug fir could be just dripping with lichens. So then you're correct if you're thinking that the FIA surveys are only looking at the tree lichens, the epiphytes. And in an ideal world, of course, we would be including terrestrial lichens and rock lichens. But we needed a way to be able to have non-experts survey because there's so many plots that need to be surveyed. And as you can probably imagine, there aren't a ton of lichenologists out there compared to some other disciplines. So I mentioned that I would be wandering around with my hand lens when I was moving through the plots. So what exactly was I looking for out there? Primarily, I was looking at these little propagules. So more vocabulary, I think the last of the main vocabulary. And these are all different propagules that you really have to know in order to differentiate species. So pay close attention here. On the left, this is the apothecium. So it's a type of ascocarp. If you know much about fungi, there are the ascomycete, the basidiomycete. It's basically cup fungi and regular fungi that we think of with real mushrooms. So these form these little cups, and they are going to give off the spores from the microbiont, from the fungus. And those little spores are going to float away and hopefully drift and land somewhere that's hospitable to them, where they will have to then recapture a photobiont partner from wherever they are. So this is the sexual reproductive structure, the apothecium. Plural, you'll hear me say apothecia. That's just the plural version of apothecium. And you can see the example down here. These little cups typically have like a shiny inner surface where the spore-bearing layer is. So then we have two different kinds of asexual propagules. We have Cerida and Isidia. Cerida are these powdery little clusters. You can see in this diagram that these little black circles are the algal cells, the photobiont cells, and they're wrapped by these little pieces of fungal hyphae. They don't have the cortex, they don't have the skin, they're just loose, like I said, powdery looking clumps of the lichen symbiosis. So that little clump will kind of erupt from a crack. As you can see here, the cortex is cracking in the top of this lichen, and those Cerida are erupting and will just kind of float away, and then they will eventually develop their own cortex and grow into a full lichen. But they're asexual because they already have all of the package of the symbiosis, right? They're both the algal photobiont and the microbiont, the fungus. When you see these cracks, you may see the word Ceralia. That is just referring to the crack where the Ceridia are erupting. So people talk about them kind of interchangeably. The Ceridia are the actual little propagules, and the Ceralia are the larger clusters of those Ceridia. So then Isidia are these little kind of more advanced versions of the Ceridia. You can see here again the algal cells wrapped up by the fungal hyphae, but they're a little more advanced because they have this layer of cortex already developed. So these little Isidia will kind of break off of the lichen thallus. You can see them down here. They'll break off, fall to the ground, or blow away, get stuck on a bird's foot, and they will hopefully land, like I said, somewhere hospitable, and then they can develop into a lichen thallus of their own, already complete with the partners that they need. My lichen professor, when I was in school, Marie Antoine, told me to, when I was looking for Isidia under the microscope, to look for tiny, shiny sausages. And that has actually remained very true. So if you're looking at them closely, you're looking for little sausages for the Isidia, little powdery clusters for the Ceridia, and then little cups for the apothecia. Here on the right are a few examples of tiny apothecia that are developing here. So the presence, absence, and morphology of these propagules vary from species to species, and you have to pay attention to them in order to recognize distinct taxa or distinct species. So when we really get into lichen identification, there are some species that can be site ID'd. You don't need microscopy, you don't need chemical spot testing, as long as you're confirming those morphologies that we talked about with a hand lens, or sometimes you can just see them with the naked eye. For specimens that cannot be site ID'd, there are several processes to narrow them down to species. So you're going to start with microscopy. You'll use both dissecting and compound scopes, depending on whether you're looking at the macro features or the micro features. Some of the macro features we've already seen, like the Isidia and the Ceridia, but I would also use the dissecting scope to make cross sections of the apothecia so that I could get at the spores. And I'll show you just what that looks like for size. This is my finger, and those are tiny fragments of an apothecium that I'm slicing up in order to get a better look at those spores. Here's what it looks like under the scope before I actually look at it through the compound scope. Very, very tiny. Very often I would be cutting these apart and like finally successfully get the best slice, breathe a sigh of relief that I finally got it, and just blow the specimen off of my microscope, and I have to start again. very frustrating. um, so you can also see here, there are some micro features that you're going to need. If you have a compound microscope, you can get at these features. That's what I was looking for when I was doing a cross section of the epithecium. Here you can make out one individual ascospore. That's the type of spore coming out of these apithecia. And you can tell that there are a bunch of them kind of jumbled into this one sac. The sac is called the ascus. And so what I would need to do is get a visual on one of these, measure one of the individual spores, talk about its morphology, and count how many ascospores are in an individual ascus. So a lot of detail. And like I said, if you're trying to ID microlichens, you have to start with the spore microscopy. Um, oh, you can even see a little algal cell down here, which is cool. So if you are unsuccessful with microscopy, if that doesn't get you quite far enough, maybe you've narrowed it down to 2 different species, but you still are not sure how to differentiate between them, you might have to resort to chemical spot testing. So chemical spot testing, again, lichens create these chemical compounds, and those chemicals, these secondary compounds, react with certain color changes when they're exposed to these chemicals that we use for testing. So you'll get a color change response, essentially. These spot tests that we use include the C test, which is essentially just undiluted commercial bleach. There is the K test, which is 10% potassium hydroxide. It's an aqueous solution of potassium hydroxide. And then there is the P test, which is p-phenylene diamine crystals. They're dissolved in 95% ethanol. So that's all super complicated. I actually have a YouTube video that goes into detail on the chemical spot testing. I made it back during COVID times for one of the Cal Poly Lichen labs, and I've just left it on YouTube. So it's about 10 minutes long, and you can watch that if you want to delve into the chemical spot testing further. Here is a picture of a tiny lichen that I'm getting a color change response on. I've just scraped off a little bit of the thallus there, dropped one of these chemicals on the little open spot, and I watched it change to red, which likely gave me a species ID, which is cool. This is a link, not a link, but just the title of my YouTube video. It's lichen identification using chemical spot testing. Botany is my account. So it's free, it's up there. Watch it if you want more detail. Sometimes even chemical spot testing does not give you all of the information that you need to get to species. You might have to resort to DNA sequencing, which I had to do for my thesis. And without getting too far in the weeds here, for fungi, which obviously includes lichens because lichens are fungi, we use the ITS region, part of the little strand of DNA, the ITS region, to differentiate between species. The reason that this works is because these are basically non-coding. So they are between two like functional ribosomal RNAs, but these are just spacers. They're non-functional RNA. So they don't have any like evolutionary pressure acting on them. So there's a huge degree of variability, even between closely related species. So once I can isolate this strand of DNA, it's usually only about a couple hundred base pairs. And I would use primers, like for instance, for ITS1 and ITS2, which is what we talk about when we say the ITS region in eukaryotes, which, plants, fungi, animals. We're referring to both these ITS regions. And so there are primers that you can use. I used ITS 1F and ITS 4 to get at the entire region. And basically, you'd put it through PCR, which is a polymerase chain reaction. It amplifies this little strand of DNA over and over and over again so that I can send it off to a lab where they have a big machine that can read the sequence from that little ITS region. And once I have that sequence, It does a really, really good job of delineating species. So it's really handy as long as there is already some kind of ITS barcode already in GenBank that you can, reference. Fungi, non-lichenized fungi, just regular fungi, are way ahead of lichens as far as ITS barcoding goes. You don't have to have access to a lab to do this. There are ways to just send off specimens. It just gets more and more expensive the fewer steps that you're doing on your own. I was doing all of my own PCR work just because I was trying to learn during my master's. But if you really get into this, there are groups where you can send uh, just your little lichen fragments and they will do the rest for you. Through my molecular work, I was able to help describe this new species of lichen. This is Hypogymnia tuckerae. Bruce McCune and I had a paper published in The Bryologist a couple years ago with its description, and we named it after Shirley Tucker, who's a female lichenologist who's currently doing incredible work for Lichenaria. And this is my collection, this photo here, and this is the type specimen for the species. I found it up on Horse Mountain. I actually collected 3 individuals up there in the Six Rivers National Forest. And Six Rivers is in the process of listing it as a threatened species. So it'll be interesting to see as we continue to monitor for it where new occurrences turn up. So this is what it kind of looks like when you're really getting into lichen identification. If you ever go out on a lichen foray, chances are there will be a field portion and a lab portion because you really just can't get very far, except, like I said, with a few species without delving into microscopy and chemical spot testing. So here I've got my lab bench. I have my dissecting scope for the first part, the compound scope to look at the spores, and then as many, keys as your heart desires. This little picture on the right, I just wanted to show you kind of the scale that I was working with. This is a little cladonia that I was spot testing. And that is how small that little lichen is. So you can you can probably see why iNaturalist and the like Seek, all of those programs aren't very good at lichen identification yet. I periodically will go on the INAT in our area in Humboldt and just knock species identification back down to genus or even family sometimes if I don't see some justification for why they've called it that species. So unfortunately, people will agree and then it's a research grade observation and it is completely incorrect, not even the right genus. So we still have a few years to go for lichens in those algorithms, but the more that we put correct lichen identifications into those programs, the better off we'll be.
Hey, Sarah, can I cut in right now? We got a question
Of
in the
course.
chat that I think is
Sure.
relevant. John Stew asks, how is the new species unique other than its DNA?
Oh, good question. Let me back up. Unfortunately, it's not some like striking morphology that's unique. The only reason that I noticed that it was different, I don't know if you can make out in this photo, but it has these kind of gray blotches all over its laminal surface, just its top of its cortex there. And they're kind of these eroded Soralia, right? Those little patches of Soredia. So it looks very similar with all of its other characteristics to a couple of different hypogymnias in the area, but they don't have these Soredia. So not everything was quite matching up with the hypogymnia that I was expecting to see. So I took this up to Bruce to say, what is going on here? Is this something new? Is it something weird? And luckily, Bruce specializes in hypogymnia. And so he was able to confirm that based just on those Soralia and the location where we were, it likely wasn't hypogymnia, oceanica or wilfii like we were thinking. Good question. And I wish it was more a more striking morphological characteristic. So once you know the basics, it gets even more fun because then you know when something is unusual. Kind of like that hypogymnia stood out to me. I was like, this isn't quite right. Something is weird. This is the largest jelly lichen I've ever seen. Jelly or gelatinous lichens take that morphology that we learned about and kind of glom it all into one single strata. So it makes it this rubbery, gelatinous thallus that really transforms when it's wet. So these were just dripping off the trees in Tanzania. And even the tribal folks that I was with who grew up in that jungle had never seen these before. Uh, it just shows that the green blur comes for everyone. Let me just show you. I'm just kind of squishing this one in my hand. Very gelatinous. Largest jelly lichen I've ever seen by far. It's some type of leptogium. And you can see how much of it is on these tree branches there. So huge lichen, 15 different people from Tanzania that I was out there with had never seen it before. Another weirdo that was really fun to find for me was down in Guyana. I was doing some mycological work in South America, and this is a weird filamentous lichen called Coenogonium. And to the untrained eye, you would just look at that and think moss. But if you know lichens and you're paying attention, you would notice all these little blobs here. Those are apothecia. Those are the sexual reproductive structures of a lichen. So even though this doesn't look like a lichen at all to me, I noticed these little things and realized that this is actually just a filamentous green algal lichen species. Really cool. Coming closer to home, another unusual thing that you'll see sometimes, this is a Lobaria anthrapsis or anthraspis. Beautiful lobaria, huge specimens here in Humboldt and Mendocino. And I noticed this one in particular because it's covered in apothecia, more than it normally would have. And this is a phenomenon that happens with lichens sometimes when they're essentially trying to abandon ship. So they don't like the thallus anymore for whatever reason. Maybe it fell off the tree and it's getting too wet, or a pathogen has come in and they need to escape the thallus. So the mycobiont, the fungus, is putting out all of these little sexual reproductive structures so that its little spores can fly away and hopefully find a more hospitable home. So sometimes you'll see these just way overproduced apothecia. It almost feels like it's masting or something. And then there are also sometimes species of fungi that will parasitize lichens, and those are called lichenicolous fungi. So those will cause weird morphologies as well that are fun to find. And another little weirdo, this mushroom is actually a lichen. This is Lichenomphalia umbellifera, and it is really bizarre. We only have two species in Humboldt and Mendocino that do this, where they, instead of evolving from the side of the fungi lineage that's the cup fungi, the ascomycetes, they evolved on the other side on the basidiomycetes. So everything is the same. They still have their algal partner, but instead of forming a little cup for its sexual reproductive structure, it forms a true, what we call a basidiocarp, a true mushroom, a true fruiting body to give off those fungal spores. And you can actually see the primary thallus, the primary body of this lichen, is this green sludge here. And so that's where the algae is. That's where the mycobiont is wrapped around the algal partner. And then when it's ready to reproduce sexually, it sends up these adorable little mushrooms. So these are fun to find. I usually find them coastal logs that have fallen, really mossy, damp areas. You can find a ton out at the dunes. A fun one to look for. Really uplifted gills that are widely spaced, and they have these little kind of depressions in the top of the cap. And if you're not sure that it's like an Omphalia, you can always just look to see if you can find that green sludge of the thallus. Another fun reason to learn all of your lichens is that you can run into genera that you recognize all over the world. So I was just out picking chanterelles in Tanzania, and I came across two lichen friends. This is heterodermia, and this is ramalina, both of which occur here in California. And when you're so far away, it really is, it's kind of just comforting to run into organisms that you know. Okay, so let's introduce y'all to a few species that you can easily identify without microscopes or without chemical spot testing or even DNA sequencing. This Usnea, this genus, is the gateway genus into lichen identification. It's always the first genus that I teach. There are several kind of pendulous, shrubby, greenish, fruticose, remember that word, branchy lichens in our area. Um, but there is one foolproof way to confirm that you're looking at Usnea. So take a look at this photo in the corner. This is what happens when you take one of the little branches of the Usnea and you just slowly pull it apart gently. The cortex will separate like it's separating here, and you'll see that there is this cord, this central cord running through every branch. It's kind of elastic, it'll be able to stretch a little bit. And if you can find that central cord in a lichen with this type of body, you know that you have Usnea, no question. It's the only genus that does that. So that's a fun way to just know one lichen genus. And then I have several Usnea species that you can ID without microscopy or chemicals. This one is one of my favorites, Usnea intermedia. Another thing I'll note here is that I don't have common names for you, and I'm sorry, there just really aren't a lot of lichen common names. And when they are used, they're broad. Like Usnea is called old man's beard, but then there are some species of Usnea that are like supposed to be old man's beard, so you can't generalize it. It's just too loosey goosey, and you just have to use the species name. So I'm sorry that I don't have cute common names for you, but they just don't really exist yet. So Usnea intermedia, you can ID this Usnea because it's the only one in our area that has apothecia. These big, wide, cup-like structures, and this it kind of looks like a brittle star or something. They always have these little projections around the outside. This individual is a huge example. That's why I took a photo. It's just like a trophy lichen. But you can see there are some smaller ones in the background, and that's normally what you would see. So if you see these apothecia on an Usnea, you know you have Usnea intermedia, no question. Another Usnea that's easy to ID and pretty coastal is Usnea rubicunda. This is a fun one because it is characterized by this reddish pigment. It looks kind of peachy to me. If you look closely, you can confirm that it has little cerealia all over the place, those little powdery patches. Um, I tend to see Usnea rubicunda on coastal scrub. Um, Often see it on alders near the coast, and it really stands out just based on its color from all the other Usnea. It's the only one that becomes this reddish color. And then finally, Usnea longissima. This is our last easy to ID Usnea. The rest of them are pretty complicated, and there's like a whole separate Usnea ID book that I use, and you have to take cross sections of the little branches and do a ratio of the cortex size. It's a whole thing. So take the Usnea species that are easy and just love them. This is Usnea longissima. It's actually the only lichen in our area that's common that has been given a rare plant ranking from the California Rare Plant Ranking System. It's a 4.2, which means that it has limited distribution. And if you look at its range, it's basically this like thin ribbon that runs along the coast right through our area. So lucky us. You can actually see this lichen as you're driving down the highway. This is one that you can ID at 65 miles an hour. And that's because Usnea longissima is the only Usnea that has these long, unbranched sections. They have, the little perpendicular branches coming off, but these main branches aren't forking very often. You get these long stretches of one single strand. So I would say if you have a strand that's unbranched, that's a foot long, you have Usnea longissima, no question. So next time you're traveling up and down the 101, see if you can see it hanging down from the conifers over the highway. Now, with lichens, you can't make assumptions, right? Remember, we're heavy on the details here. So you might look at this now and be like, oh, more Usnea, perfect. But actually, this is mostly called Alectoria. Um, and if you don't pay close attention to all the structures, you'll miss beautiful Alectoria lata. It has these little moon-like apothecia. I didn't find it very often on Horse Mountain. There was a ton of like Alectoria sarmentosa, this big stranded one. But if I looked more closely, sometimes Alectoria lata would pop out. They have these really cool, they're called pruina. They're little crystals, calcium carbonate crystals that develop on the surface of the apothecia. And they kind of function as a sort of sunscreen, sun protection, we think. They're beautiful. So keep an eye out for Alectoria lata. Here is another Lost Coast classic species for you. This is Ramalina menziesii. This has a common name. It's called lace lichen. In fact, it's also our state lichen. Do you know we had a state lichen? It's Ramalina menziesii. This one is also pendulous, so it looks a lot like this Alectoria. But again, if you get up close, you're looking at the details, you can find that there are these little nets. That's why it's called lace lichen. These tiny nets where the branches kind of fork and go back into each other. Sometimes they're really, really small. But if you can find one of these little nets, no question, you have Ramalina menziesii. This one is usually like pretty much on the immediate coast and is in big pendulous swaths like this. This was just at the Samoa Dunes in Eureka. Another adorable little species that you will find on the Lost Coast. It may not look like very much, but it is fun. It's characterized by these large white Soralia. You can't see them very well in this picture. You would need a hand lens. You don't need a microscope, but you would need a hand lens. The branches kind of terminate in these Soralia that are spilling out those little powdery white reproductive structures, right? My favorite thing about Nibla, this little species, is that it only grows within sight or sound of the ocean. So hypercoastal species. It also does a really weird thing when it's stored as a collection. They develop these like spider web looking crystallized tufts in between the branches. So it just looks like a spider has kind of invaded them if you leave it in a drawer for a few weeks. So that's another way to confirm the species if you're, not in a hurry and you can just store it for a little while. Tiny little thing. You can, my finger is right here in the bottom corner. It's itty bitty. Looks a lot like Ramalina, but again, if you can confirm the end of the branches have those Soralia and you're right next to the ocean, you probably have Nibla. Platysmacia glauca is a super common species of lichen. This one actually also has a common name. It's called tattered rag for obvious reasons. It just looks like a tattered rag. It looks very messy. It's extremely plastic in its morphology, so it has a lot of various looks to the tattered rag. But if you're paying close attention, you can find the more exciting Platysmacia wheeleri. Again, you don't need a microscope. The only thing you need to see are these dramatic undulating margins that are covered in Ceridia. You can see the kind of powdery texture on the ends there. And if you see these big dramatic waves and it's a Platysmacia, this is Platysmacia wheeleri. The only difference between the two are these margins. This is a fun treasure hunt lichen. You really have to be looking. But again, you don't need a microscope. If you find them, you find them. You can tell how tiny they are by these moss leaves in the photo. Each one of these apothecia is only, a couple millimeters across. But if you find squamulose lichen like this in the moss that has these beautiful little kind of pearl-like projections around each apothecium, you have Seroma hypnorum. Squamulose, adorable, one of my favorites. Very small. Here is yet another example of an exciting species that you would miss if you weren't paying close attention. There is a common species of Umbilicaria. It's called Umbilicaria fea, and it looks identical to this species, except the apothecia are round. If you've spent any time on local rocky outcrops, you'll likely find Umbilicaria. And if you're looking very thoroughly, you might find them with these super angular apothecia, which is why it is Umbilicaria angulata. These apothecia are super cool. You can tell that they have all these extra folds in them, and that's basically just to increase the spore dispersal surface area, right? So just like gills of a mushroom, they want to have more surface area for spore production. That's exactly what these little apothecia are doing. And just for size scale, you can see this is what the specimen looks like when it's in my hand. So this is the same specimen in the background, just the big thallus. And you'll see these, like I said, it looks identical to this all over rocky outcrops, except all of the little apothecia are round. So keep an eye out for the fun angled versions. Umbilicaria angulata. And last but not least, the poster child of lichens. This is Lotharia columbiana, this yellow one here. It's called wolf lichen, although they kind of call all the Lotharias wolf lichen. Unfortunately, it has that name because back in settler times, they would stuff this lichen into pieces of meat and feed it to wolves and foxes who were coming after their livestock in order to poison them because this lichen has its own chemical. It's called volpinic acid. And they would poison wolves and foxes with it, sometimes even putting broken glass in the meat. It's awful. There are several Lotharia species in our area. We probably have a species complex, but the only one that has these beautiful apothecia with the little kind of sunshine projections is Lotharia columbiana. So as long as you have a bright yellow lichen that's fruticose, not foliose, you want it to be branched, not flattened, and it has these brown apothecia with the little sunshine projections, you know you have Lotharia columbiana. These will typically be more inland. They like higher elevation. I saw a ton of these on Horse Mountain, for example. Okay, so now I know that all of you are just dying to get out there and ID lichen species. So I have a couple of recommendations for you. For this area, Humboldt and Mendocino, my favorite ID book is Macrolichens of the Pacific Northwest by Bruce McCune and Linda Geiser. It has an excellent introduction that covers a lot of the basics that we talked about today. And it also has color photos and descriptions with spot test results. And it also has an illustrated glossary, which is really, really helpful for just starting out. Bruce McCune also has two gorgeous, I've got them right here, two gorgeous volumes of microlichen books, if you're interested. They're incredible. If you really fall down the rabbit hole and you want to get into microlichens, you're going to need Bruce's microlichen volumes. I also recommend having some form of hand lens while you're out there. And I really like the Bausch and Lomb. It's called the Hastings Triplet, the Bausch and Lomb Hastings Triplet. But you can find cheaper ones, $10 a pair. These are probably 25 or 30. I use a 14X and a 20X for my really tiny specimens, but you could probably get by with 10X. I also, lastly, I always feel like I need to remind people that collecting lichens is not quite like collecting fungi or berries or flowers. When you pick a mushroom or a flower, you're essentially just picking an apple off of a tree, right? You're just taking the fruiting body. But when you pluck a lichen, that's the entire lichen, it's the entire lichen thallus. So some species are incredibly slow growing. They can be hundreds of years old, even if they're really small. So there's plenty of lichen litter on the ground. You don't need to collect it off the trees or rocks unless you're doing serious research, in my opinion. And you don't want to collect more than is necessary for identification. Just remember that they are crucial part of healthy ecosystems. Um, and if you're collecting lichens, remember it's way easier than collecting fungi. You can just stuff them into little paper packets. You don't have to mess with silica and drying things or pressing them into herbaria sheets. You can just throw them in a paper packet, toss them in a drawer. 50 years from now, you can pull them back out, drop some water on them. They'll rehydrate and be just fine. It's pretty remarkable. They're very low maintenance. And remember, while other organisms have temporary seasons, lichens are around all year long and they don't, fly or run away. So if you're used to collecting other specimens, lichen collecting is really a dream. Um, okay, so I know we have barely scratched the surface today. We're out of time, but I hope I gave you something to start with, something to go on. And if you find yourself wanting to know more about lichens, I recommend following a few social media accounts. I'm not really on there anymore these days, but occasionally, I'll be sharing all my lichens from Africa here soon. And I'm at Hum Botany on Instagram. And then there is California Lichens, which is the California Lichen Society, and Northwest Lichenologists, that's the Pacific Northwest group. And they both hold forays and lab days where you can get together. They host lectures. Highly recommend following both of those accounts and just really diving in to the wonderful world of lichens. Thank you all so much for your time.
On behalf of Friends of the Lost Coast and the Bureau of Land Management King Range National Conservation Area, thank you for tuning in to Lost Coast Lectures. To view the complete archive of recordings from this series, or to learn more about our work inspiring a passion for nature, visit Friends of the Lost Coast online at lostcoast.org.